Hello friends, we are going to discuss some important biochemistry MCQ which were asked in Neet PG 2022. There are about 12 MCQ are asked based on the biochemistry and they are mostly belong to the topics of vitamins, minerals, genetics and metabolism of carbohydrate, lipid and protein. So one by one each MCQ we are going to look and their option and right option that we are going to select and how to select that right option that we are going to discuss. So first MCQ is about a patient from a Mediterranean country visits Africa where he develops malaria. He is treated with primaquine and later develops hemolytic anemia. Deficiency of enzyme involved in which of the following pathways could be the cause. So we have to first identify deficiency of enzyme involved, but here not a name or not a single name of enzyme is given, but related to which pathway that we have to identify. Now already you are knowing about glycolysis, HMP shunt pathway, gluconeogenesis, Lubrin-Rappaport pathway. Now Lubrin-Rappaport pathway that is related with the 2,3-bisphosphoglycerate, that is 2,3-BPG. Now, when we are talking about hemolytic anemia topic that comes in relation to this all pathway, out of that glycolysis and HMP shunt pathway that is somewhat related with the hemolytic anemia. Gluconeogenesis that is not related with the hemolytic anemia. 2,3-BPG or Rappaport Lubrin pathway, shunt pathway that is also not related with the hemolytic anemia. So directly two options 3 and 4 that is rule out. Only two options are remaining, glycolysis and HMP shunt pathway. Now further option is given malaria and primaquine. Now this malaria and primaquine that is typically related with the HMP shunt pathway. Glycolysis in that typically pyruvate kinase or hexokinase enzyme deficiency that maybe look like a condition that is sometime disorder occur with the glycolysis, but in that malaria and primaquine is not mentioning. So basically why this HMP shunt pathway, that also we are going to discuss. HMP shunt pathway that are oxidative phase and non-oxidative phase. Oxidative phase, simply you have to remember HMP shunt pathway oxidative phase is required for the formation of NADPH, and this NADPH is required to maintain red blood cell membrane integrity. Now when this NADPH is not present, stress level which is generated due to the drug intake, that is primaquine, and that stress that will lead to the destruction of the red blood cell and that will lead to the hemolytic anemia. So here definitely answer that is a G6PD deficiency. In that patient of G6PD deficiency, if primaquine like drug is given, and this drug is typically given in malaria, and if this drug is given, that will lead to the stressful condition and that will lead to the destruction of RBC and hemolytic anemia will be there. So here definitely answer related to the G6PD enzyme deficiency, and G6PD is required for the HMP shunt pathway, particularly oxidative phase. So here the right answer that is HMP shunt pathway. So deficiency of enzyme that is G6PD involved in which of the following pathway, that is related with the HMP shunt pathway, which is also known as a pentose phosphate pathway. Now another MCQ, which of the following helps in the transport of fatty acid across the inner mitochondrial membrane? Now you have to remember that beta oxidation of fatty acid that is typically occurring inside the mitochondria, means matrix part of the mitochondria. Now this fatty acid need to be transported across the cell inner membrane as well as outer membrane of the mitochondria. Now inner membrane of mitochondria that is not permeable for this fatty acid, so fatty acid need to be transported, and that transportation is occurring with the help of particularly two protein CAT1 and CAT2, as well as in between that two particular protein is present, that is translocase. Now this CAT1, what is the full form of CAT? That is carnitine acyl transferase. So first answer that is carnitine, that is most likely the right answer. Other options like acyl carrier protein, that is one part of fatty acid synthesis. Fatty acid synthesis, in that one word is there ACP. ACP means acyl carrier protein, but it is a carrier but not helping for the transportation of fatty acid for inner mitochondrial membrane. Lecithin cholesterol acyl transferase, that is LCAT, which is required for the transfer of lecithin, and in that is reaction, metabolic reaction between lecithin and cholesterol, that is acyl, means fatty acid is transferred between lecithin and cholesterol, and that is the function of LCAT. It is not for the transportation of fatty acid across the inner mitochondrial membrane. Carnitine and albumin, basically carnitine and albumin is not the answer, because albumin is transporting fatty acid in the blood. So albumin is required for the transportation of fatty acid in the blood, but not the required for the inner mitochondrial membrane. So right answer here that is carnitine only, and that is the right answer. Now further another MCQ, an adolescent male patient presents to you with exercise intolerance. He gives a history of developing cramps on exertion. Which of the following enzyme deficiency could be the cause? Now for the biochemistry you have to remember one particular exercise intolerance and muscular cramps, and these two words that is typically related with the muscle phosphorylase enzyme deficiency. And for that muscle phosphorylase enzyme deficiency you have to remember McArdle's disease. So there are different glycogen storage disease, and for the glycogen storage disease, that all the name and which enzyme deficiency that you have to remember. Here some hint is given, exercise intolerance and muscle cramp, that is typical hint is given in the question, and from that you can directly jump that M, that exercise intolerance related to the muscles, and that is enzyme deficiency, that is myophosphorylase. It can be written as a muscles phosphorylase enzyme deficiency, glycogen phosphorylase enzyme deficiency, or McArdle's disease. Now, related to the same, there are another disease, that is Hirsch's disease, where hepatic phosphorylase enzyme deficiency. Now, other ABCD that you have to remember for the glycogen storage disease: A for the Anderson's disease, where enzyme deficiency, that is branching enzyme. C means Cory's disease, where D means there is a D branching enzyme deficiency. Now, out of all these, particularly muscle cramps as well as exercise intolerance, this word is typically related with the McArdle's disease or the muscle phosphorylase enzyme deficiency, muscles, which is also called the myo. So that is myophosphorylase enzyme deficiency. That is the right answer in this MCQ. So right answer is myophosphorylase or muscle phosphorylase enzyme deficiency. Now, next question, that is based on genetics. A single mutation in a nucleotide base pair resulting in a termination codon is known as, means a mutation is occurring and in nucleotide base pair, that is lead to the now formation of the termination codon. So basically this termination codon, that is formed as a mutation. Now there are different types of mutation. There is a silent mutation, missense mutation, and nonsense mutation. Stop mutation, that is not a, that is mutation, but it is not considered as a type of mutation. So silent mutation, where there is nothing difference occurring after the mutation, whatever the mutation is occurring, but it is not reflecting anything, any change in the particular. Suppose that there is a one amino acid and for that different genetic codons are there, so that change in genetic codon, but that mutation led to the change in genetic codon, but that amino acid is remain the same. So that is silent mutation, and that is not creating any problem. Now another, that is missense mutation, that is also of a three variety: acceptable, partially acceptable, and non-acceptable. So missense mutation, in that what happen, acceptable means there is a change is occurring, but that is not a making any difference. Suppose that aspartic acid is replaced by the glutamic acid, or glutamic acid is replaced by the aspartic acid, that is not making any much difference, so acceptable, or that is not making any change. Now another, that is partially acceptable, means suppose that mutation is creating problem but also giving some advantage, but both the things are occurring, means occurring, but that is also creating some problem and giving some advantage, like sickle cell anemia, that is giving one advantage, but one advantage that against the malaria falciparum infection is not seen in a patient of sickle cell anemia. But oppositely, there are lots of problems related to the sickle cell anemia. So it is a partially acceptable. Not acceptable means that whatever the protein changes is occurring, that is not acceptable, or functionally inactive protein is generating, which is not compatible with the life, and that is called a non-acceptable mutation. So missense mutation, that is also of three variety. Now nonsense mutation, here the mutation is related with the terminator codon. So terminator codon, so that is generated as a part of mutation, or sometime what happen, that mutation is leading to the change of terminator codon. From that terminator codon, it is now changing into the coding codon, means that is now changed to the making of giving signal for the one amino acid. So here the basic thing is that single mutation in a nucleotide base pair resulting in a terminator codon is known as, so change is occurring in a terminator codon, so you have to select the right option, that is nonsense mutation. So silent mutation is not an option because the word termination codon is utilized, so base pair resulting in a termination codon is generating, so this terminator codon formation, that is nonsense. Now in this kind of mutation, what happen, whatever protein formation, these protein formation, suppose of 100 amino acid, now termination codon, that is from the 75th amino acid, now from that protein will not be synthesized. So whatever the protein is synthesized, that is of no use, or it is not making any sense for the protein for functionality, so it is a nonsense mutation. So it is basically, the right answer is nonsense mutation. Now DNA packing is done by which of the following? So here glycoprotein, histone, nucleic acid, and adenine. So all options are given, but typically you are knowing that nucleoprotein, that is typically histone. So when DNA is generated, this DNA will wrap around this histone protein. Histone protein are also of a different variety, but out of that H2A, H2B, H1, and out of that this octamer protein, that is histone, and around that DNA will be wrapped and that will make a nucleosome. So you have to remember which protein is required, that is histone. Now again, that histone protein is also rich in a negatively charged, means that was that particular arginine and lysine that is predominantly found in this particular histone protein, and that is making a different kind of charge. So arginine and lysine, which is containing NH3 that is now converted into the NH3 plus, which is having more amino group, and this more amino group that is mostly having a positive charge, and that can easily attach over the negatively charged DNA molecule. So histone is the right answer. Instead of that, sometime it is written which amino acid is predominantly found over the histone, that is lysine and arginine. So you have to select the right answer, that is histone, which is required to make a nucleosome, and that is further condensed to make chromatin and chromatin fiber. Now another MCQ, which of the following enzyme activities can be estimated in red blood cell to diagnose vitamin B2 deficiency? Now here the some typical theory question are there. For the some vitamins you have to remember which enzymatic activity is measured. Suppose for the vitamin B1, transketolase enzyme activity is measured. Vitamin B2, that is glutathione reductase enzyme activity is measured in the red blood cell. For the kynureninase, that is measured for the vitamin B6 enzyme deficiency. So this is the typical question and answer, typical theory part. Transketolase, that is also this enzyme activity requires vitamin B1. Glutathione reductase, that is basically related with the free radical scavenging system, but glutathione reductase is measured as a part of deficiency of the vitamin B2. Now kynureninase, that is part of the metabolism of this tryptophan, and in this metabolism you have to remember vitamin B6 is required for the activity of kynureninase, and this enzyme is required for the vitamin B3 formation. Now pyruvate dehydrogenase, that is multi-enzyme complex which is requiring 3 enzyme and 5 coenzyme, so that is another thing. But vitamin B2 deficiency, for that you have to remember glutathione reductase enzyme is measured in red blood cells. Now another MCQ, the following is due to the deficiency of your typical picture of Bitot spot, and this Bitot spot, that is simply you are knowing that vitamin A deficiency. Vitamin A deficiency, there is xerophthalmia, keratomalacia, Bitot spot, night blindness, all are the typical symptoms of the vitamin A deficiency. Vitamin C scurvy, vitamin B complex, there are so many vitamins and so many disorder. Vitamin E, that is mostly related with the antioxidant activity. Generally deficiency is not seen, but vitamin A you have to remember particular Bitot spot. Vitamin E you can remember that one more thing, that is selenium mineral. So here the right answer is vitamin A. Now here interesting question, a child present to you with anemia, thrombocytopenia and bony pain. On examination, hepatosplenomegaly was noticed. If light microscopy of bone marrow aspirate revealed crumpled tissue paper appearance. This word crumpled tissue paper, that you have to keep in your mind, that you have to permanently remember, that this word is typically giving you idea about the one disorder, that is Gaucher's disease. What is the enzyme defect? It may be written as a name of disease, or it may be written as a enzyme deficiency. So glucocerebrosidase, that is the enzyme deficiency in Gaucher's disease. So Gaucher's disease, glucocerebrosidase enzyme deficiency, which is creating this crumpled tissue paper appearance. So this typical cell appearance will be seen, and so right answer is glucocerebrosidase, and that is occurring in a Gaucher's disease. Hexosaminidase, that is related with the Tay-Sachs disease. Sphingomyelinase, that is related with the Niemann-Pick disease. Glucose-6-phosphatase enzyme deficiency, that is related with the von Gerth disease. But here the typical anemia is written, bony pain is written, hepatosplenomegaly is written. So hepatosplenomegaly is not seen in a hexosaminidase. That is also one point you have to remember. Glucose-6-phosphatase, that is leading to the hypoglycemia. But here typical anemia word. Anemia is typically observed in this Gaucher's disease of glucocerebrosidase enzyme deficiency. And in all these conditions, this crumpled tissue paper appearance is not seen, only it is seen in a glucocerebrosidase enzyme deficiency. That is Gaucher's disease, right answer. So basically hint are crumpled tissue paper cell, anemia, bony pain and hepatosplenomegaly, all are going to the only one answer, that is glucocerebrosidase enzyme deficiency. Now another question, the electron transport chain is a series of a redox reaction that results in ATP synthesis. Now that is common sentence, which of the following is a cytochrome complex 4 inhibitor? So cytochrome complex 4, that the typical complex inhibitor is asked. You have to remember all inhibitors, that is H2S, that is hydrogen sulfide gas, carbon monoxide and cyanide. These are the typical inhibitor of complex 4. Here carbon dioxide is written, not a carbon monoxide. Cyanide, that is the right answer. Oligomycin, that is inhibitor of FO portion of the complex 5, not complex 4. Complex 4 is not related with that oligomycin. Oligomycin is related with the complex 5, have a 2 portion FO and F1. Out of that, FO is inhibited by oligomycin. Oabin, that is inhibitor of sodium potassium ATPase pump. So all are their option, but out of that right answer is cyanide, which is typically complex 4 inhibitor. You have to remember H2S gas as well as carbon monoxide that are also inhibitor of the complex 4 of the electron transport chain. So electron transport chain inhibitor are also important. Now, another interesting question. A paint factory worker working since 2 years complain of joint pain, insomnia, fatigue, headache. He is found to have anemia. Enzyme affected is. Now, here options are given like UPG3 co-synthase, ferrochelatase, UPG1 synthase and ALA synthase. All are related with the heme synthesis pathway. So, from the option you can now choose one pathway, that is heme synthesis. In heme synthesis, that some all are the important, all are related with the some porphyria, but nothing about porphyria. Only thing is about anemia. And related to the anemia, you have to remember a paint factory, that is the hint. Paint factory basically utilizing one mineral, that is lead. Lead poisoning, that is typically related with the paint factory. So lead, that one topic related to the biochemistry. In that paint basically containing lead is basically inhibiting ion absorption, that is one thing. Once it is present inside the body, that is inhibiting two particular enzyme for the heme synthesis, ALA dehydratase, not ALA synthase. That is ALA synthase first enzyme, but it is not inhibited by lead. Lead is inhibiting ALA dehydratase. And another enzyme which is inhibited by lead, that is ferrochelatase, which is also called as the heme synthase. So second enzyme of this heme synthesis and last enzyme of heme synthesis, that is ALA dehydratase and ferrochelatase, which is inhibited by the lead, which is present in paint. Okay, so from the paint factory, another hint, that is anemia, and all symptoms of the anemia all are going to the particular answer, that is ferrochelatase enzyme deficiency, or that is affected in this condition. So ferrochelatase is the right answer in this condition. Now another MCQ, a woman on antidepressant present with bleeding. Now bleeding word is there, you have to catch which vitamin is related with the bleeding. So definitely vitamin C is there for the bleeding, also scurvy. Another, that is vitamin K from the fat soluble vitamin. So she gives a history of bulky stool. Which of the following vitamin deficiency can cause bleeding in this condition? Now bulky stool as well as that bleeding and antidepressant all are related with some typical situation that fat absorption is affected. But another thing, that is bulky stool and antidepressant, that is not making much related with the fat, but option are like vitamin A, D, E and K, all are related with the fat soluble vitamin. Now out of that fat soluble vitamin, typically which one is related with the bleeding disorder? So typically vitamin K, which is related with the bleeding disorder. Another thing, that is antidepressant and this bulky stool, that is also giving some hint, some metabolic disorder related to the fat soluble vitamin. So all are going to the right answer, only vitamin K, which is affecting factor 2, 7, 9, 10. Vitamin A related with the vision, skin and mucous membrane. Vitamin D related with the bone structure, that is osteoporosis and as well as rickets. Vitamin E mostly related with the antioxidant activity, but here the right option, that is vitamin K, which is affecting factor 2, 7, 9, 10 by gamma carboxylation of glutamic acid, that is vitamin K cycle. Now another MCQ, last MCQ of this series, that is a known patient of tuberculosis on INH for the past 3 months. So INH, that is isoniazid, that is one drug which is given in a patient of tuberculosis, which is given for the last 3 months. Complain of numbness, tingling and burning sensation in all the extremities. What could be the vitamin deficiency? Now these are the some particular word which is related with the some particular vitamins as well as minerals. So INI, that is isoniazid, which is given, and here typically when vitamin B6 you are reading, this drug is definitely there. So pyridoxal phosphate, which is INH is structurally similar to the pyridoxal phosphate, and that's why deficiency of vitamin B6 will be there. Now B6 is basically required for the protein metabolism, and its deficiency that can lead to the all disorder related to the vitamin B6 activity inside the or B6 related reaction inside our body. So numbness, tingling, burning sensation all are related with the somewhat CNS involvement, but B6 deficiency that is also related with the formation of different neurotransmitter, and B6 deficiency that can also affect the protein metabolism. Now here other options like B3 deficiency, but B3 basically related with the niacin deficiency, B2 riboflavin, B1 that is thiamine deficiency. But here symptoms are somewhat related to the CNS, and when the particular word INH is written, then you don't have to choose any other option. You have to go with the particular vitamin deficiency, that is vitamin B6. Now suppose INH is not given and subacute combined degeneration is written, then you have to choose vitamin B12. Further, if vitamin B1, there is a chronic alcoholic patient is written and there is all symptoms of CNS, then you have to change the option, that is vitamin B1. Now all the condition, like if there is a chronic this antibiotic is taken, that our intestinal bacterial flora is affected, and that is also making that angular stomatitis, glossitis, ileosis, then you have to choose the vitamin B2 as a option. So from the question, that will give the hint, and from the hint you have to choose the right option. So here the right answer is vitamin B6 deficiency. So that's all about all MCQ which were asked in NEET PG 2022.